Okay, so to help me welcome Dr. Oclarit, may I ask our CEO introduce Dr. Oclarit?
Welcome, everyone. This is Susan Barlian, but anyway, please help me welcome Dr. Oclarit to give us a lecture on genomics and DNA and its value to healthcare. Dr. Oclarit, good morning.
Good morning,
Good
Dr. Oclarit.
morning. If we talk about genomics, we cannot help but talk also about proteomics because the genes become proteins. And how these genomics are engineered, that's why we cannot help but discuss also the aspect on genetic engineering. Genomics is made possible because of the advent of the field called genetic engineering. Okay, before I begin, I would like to define some terminologies. When you talk about DNA, DNA is the overall design of the genomics because the DNA are the materials for the genome. And the DNA is composed of mononucleotide. One mononucleotide is composed of three components: the sugar pentose, the phosphoric group, and the nitrogenous base. The three components, when connected together by a covalent bond, they become what you call mononucleotide. One mononucleotide is connected to another mononucleotide in a backbone called phosphate sugar backbone. So the underlying chemical bond between one mononucleotide with another mononucleotide is called phosphodiester bond. So you will notice there that phosphoric acid is always needed, no? So that is what you call DNA. So the DNA is just a chain of nucleotides. And then that is the DNA. So when the chain of nucleotide is converted into a protein, that particular chain of nucleotide is known as genes. So the sequence of mononucleotide, which are DNA, and that sequence, that code for a protein is called genes. And the collection of several genes in a particular human body is called as genome. So the total number of genes in your body is actually the total genome of a human person, no, of a human body. There now, the term emerged what you call genomics. So when you say genomics, then it refers to the DNA sequence that are coded into proteins. Those are what you call, again, is called genes. Why is it that we have to differentiate the DNA which is gene and the DNA which is not gene? There are DNA sequences that are not genes because they are what you call introns. Introns are just DNA sequence that are blank. There are no equivalent protein for their codes. The sequence that are nonsense, and you know there are 90% of our DNA sequences are nonsense. So 90% of our DNA sequences are introns. So those genes that are coding for protein, the term for that is technically what you call exons. So exons are genomes. Exons are equivalent to the genes. And the introns are the nonsense code. The nonsense code are just mere sequence of DNA that have no equivalent protein for their sequence. Whereas the gene is a DNA sequence that has an equivalent protein for that particular code. And so that's why the term proteomics. So if you talk about genomics, which are genes or sequence of DNA that codes for protein, that is genomics. So if you talk about genomics, you cannot help but talk about proteomics because the genes are converted into proteins. And that particular genes could be engineered in order to produce a new protein as desired in the process of medical intervention or in the process of changing the expression of the protein requires genetic engineering, either natural genetic engineering by our own body or an artificial genetic engineering in a particular technology that is available nowadays. Okay, let's proceed. So genomics is an emerging field that deals with the DNA sequence that has an organization and function to make protein. So And then there is the evolution of genomes, because some genomes are engineered to become new genome in order to change the expression of that gene and change the expression of the protein. So as what I said, genomics will result through what you call the field called proteomics. Proteomics refers to the protein that is engineered. Proteomics aims to identify all the proteins in a cell or organism, including any post-translationally modified proteins, as well as their cellular localization, functions, and interactions. Because proteins are mere sequence of amino acids. Mere sequence of amino acid does not mean functionality immediately. It has to be modified, what you call post-translational modification. That happens in the endoplasmic reticulum of the cell of the body. So the endoplasmic reticulum of our cell is always associated with ribosomes, because as soon as the protein or as soon as the genome is translated into protein, they are actually entered into the endoplasmic reticulum for post-translational modification, meaning the sequence of amino acids are modified. One modification is what you call folding of the protein to configure into a particular shape and functionality. Therefore, post-translational step in the proteomics, which is actually a result of genomics, is actually very necessary for the protein to become functional. So proteins are not functional until they are modified in the endoplasmic reticulum. And that modification requires enzymes, requires epigenetic factors. So modification is very important. That's why in our supplementation immunogen is not only nucleotide, but it also consists of some proteins which are considered to be modifiers and considered to be important in post-translational modification in order to rightly configure the protein to become a functional protein. And remember this, according to the study, genomics started in, I think, 1988 or 1989, and it ended up in 2003. And they found out that in the human body, there are about 30,000 genes. So these 30,000 genes comprise now what we call the genomics of the human body. So we have about 30,000 genes in the human body. And remember that gene is converted into protein. One gene equals one protein. So if you want to modify the protein, you have to modify it at the genomic level. Meaning to say that if you want to change the expression of the protein, you have first to change the sequence of the genomes in the DNA comprising the genes. As I repeat the definition of the gene, the definition of a gene is that they are DNA sequences that are converted into proteins, because there are DNA sequences in our body that are not converted into protein. They are called introns. So we are talking now about the genomics. So we are talking about the exon, or the set of DNA codes that has the ability to be transcribed first into RNA and then translated finally into protein. And so this is the field of genomics. While genomics was made possible, the arrangement of the gene or the modification of the gene either artificially or naturally is made possible by what you call the recombinant DNA technology. It is also known as gene cloning or genetic engineering. So nowadays, you can actually isolate a particular gene and engineer it and reintroduce it to the body and find out the effect of that particular engineering. So genomics is actually a field in science that is emerging today, where a particular gene is engineered in order to change the expression of that gene in order to protect you from possible damage or possible physiologic, what you call this, defect. So early detection of a defective genomics will be very useful in medical therapeutics. So for example, Down syndrome. If Down syndrome is detected earlier, then you can engineer the genomes and rearrange it, remove the gene that are not good. So the field in genomics make possible the splicing or the removal of the sequence of DNA that are defective, and then resynthesize it, rearrange the nucleotides, and reintroduce that particular engineered genome back to the cell and express it into a protein. So this is now a field in genomics. So it is now made possible, supposing you have a genetic disease like G6PD or glucose 6-phosphate deficiency enzyme deficiency. If that deficiency is detected earlier, then you can introduce the correct genome into the cell, and then finally, the defect is corrected by inserting the gene into the site where it was damaged. So it is nowadays, it's now possible to identify the gene, sequence it, and if the gene sequence is defective, remove it out. To remove the defective gene or to remove the defective nucleotide, you have to use the enzyme what you call restriction enzyme nuclease. There is an enzyme available in the market today where you can cut the DNA from one site to another site. If you have identified the defective sequence, then you can cut it from one site and to another site, and then you can insert a new sequence to that site in order to correct the error. So that is what you call DNA-based medicine. So genomics is a DNA-based medicine. It is an emerging field where genes are rearranged, where genes are modified, where defective genes can be removed from the site and then reintroduce the gene into the area in order to express the correct protein which was damaged. So that is the field in genomics. Therefore, the field in genomics was made possible because of the invention of the recombinant DNA technology. This recombinant DNA technology is in the field of genetic engineering. It utilizes the enzyme called restriction enzymes. Restriction enzymes are just like scissors that will cut one part of the DNA in order to remove the defective. So one of the most important aspects in genomics is for our ability to identify the sequence. Because nowadays, there are already machines that are able to sequence the DNA. I was able to experience that. I myself was able to sequence some genes way back in 1996 in Japan. So I am very familiar with how these particular genes are engineered. So first of all, you have to sequence the DNA. So you need DNA sequencer machine, which is now available in the market. So a DNA sequencing machine costs about 30 million pesos. So if you have money, you can buy that machine and you can extract the blood and then get the DNA and sequence it in that DNA sequencer. And then you will know the sequence of your nucleotides. And from there, you will know which sequence is defective and which sequence is not. So the field in genomics is made possible because of the available technology, which is actually in recombinant DNA that requires gene sequencer machine. It requires an enzyme called restriction enzyme that will be able to cut the DNA at a certain desired sites or at certain desired point where the defect is found. So in genetic engineering, the immediate goal of the experiment is to insert a particular fragment of the DNA into a particular vector. And the favorite vector for inserting a fragment of DNA is what you call plasmid. So plasmids are extra chromosomal unit of the bacteria that is capable of carrying a fragment of DNA of interest. For example, there is a person that has diabetes. So because that person requires an insulin, so what we will do is get a DNA for insulin and insert the plasmid into the bacteria and make that bacteria or enable for the bacteria to produce insulin from the inserted DNA. So in genomics, what is important in genomics is actually to isolate the DNA of interest. And then that DNA of interest is inserted into the plasmid, and that plasmid is inserted into the bacteria, and the bacteria is cultured, and the bacteria will now manufacture human insulin, because although bacteria is a bacteria, but that bacteria that has been inserted with a human DNA for insulin, that bacteria is now capable of producing insulin. So you can harvest the insulin and inject it to the person that has a defective pancreas. So that is now made possible because of genetic engineering and genomics. So one of the carriers of the fragment of the DNA is the virus. Actually, viral DNA could be a vehicle to insert the DNA of interest. So if you have identified a gene already, you can actually insert it into the virus and inject the virus into the area. The virus will now insert the gene into the area of damage. So the virus is capable of inserting a particular fragment of engineered genomics or the engineered genome. So this is accomplished by breaking the DNA molecules at the specific sites and isolate particular DNA fragments. That DNA fragments are then usually obtained by the treatment called restriction enzymes. This is the one I told you earlier. So that fragment you have identified can now be inserted into the area using the enzyme called restriction enzymes. So cloning from messenger RNA molecule depends on unusual polymerase. We called it reverse transcriptase, because inside our cells, there are enzymes that are able to revert messenger RNA. So an RNA molecule can be reverted back to DNA using the enzyme called reverse transcriptase, which can use a single-stranded RNA molecule as template and then synthesize a DNA from RNA. So that is called reverse transcription. And from RNA into DNA, you can make actually a complementary DNA. You call it the cDNA. So many of the products of genomics today are library of cDNA. So actually, it is now possible to construct a cDNA for all the possible proteins in our bodies. So in genomics, they aim now to construct a cDNA for every protein in the body. Remember that proteins are sequence of amino acids. And sequence of amino acids are coded from the codon. And the codon is an RNA. And that codon RNA could be reverted back to DNA using the enzyme restriction enzyme. In other words, if you know the sequence of the protein, or if you know the sequence of the amino acid in that particular protein, you will be able to construct an mRNA from that particular protein and then revert it back to DNA. So for example, insulin gene or insulin DNA. So you make a reverse transcriptase and then you make a complementary DNA for insulin. So it is now possible to make all proteins in our body could now be constructed into cDNA using the reverse transcriptase. So you can make a library of all proteins in your body. So if you can make a library of all proteins in your body using the cDNA technology, then any protein that is defective in your body can now be reconstructed using a cDNA, and use that cDNA of that protein and insert it back to the body using the plasmid and using the viral DNA as vector. So it's now make possible that if you have a particular protein, you can construct a cDNA. So a genomic is actually the reconstruction of the cDNA, or C stands for complementary DNA, because the cDNA is actually derived from the RNA molecule using the reverse transcriptase. So it is now possible, if you know the sequence of the amino acids of that particular protein, you can revert it back to a codon, because one amino acid is coded by a codon, and the codon is a messenger RNA, and that messenger RNA can be reverted back to DNA. That's why every protein has a messenger RNA, and that messenger RNA, when reverted back to DNA, is called cDNA, or complementary DNA. So this is how they do the cloning of the messenger RNA. The cDNA cloning, the resulting full length of the complementary DNA contains an uninterrupted sequence for the protein of interest, because ordinarily the messenger RNA is actually interrupted with introns. That's why in the cDNA, there is no more interruption. In the natural way, there is always the interruption. That's why there is a need for splicing the RNA prior to protein synthesis in order to remove the nonsense codes or remove the introns. So the natural RNA molecule has introns, but the cDNA has no more introns. Therefore, it can already be directly inserted into the cell where the defect is, and then you can express that cDNA into a protein, and finally restore the function of that particular protein in order to restore the particular function of that particular DNA molecule. So if the DNA sequence is known at both ends, the complementary DNA for design or appropriate primers, and then you can magnify. So in genomics, you can actually multiply the gene already if you know the sequence of the DNA from one end to the other end. If you know the sequence of the gene from one end to the other end, you can construct what you call a primer, and that primer can now be used in order to magnify the molecule of the cDNA using the enzyme called reverse transcriptase in the PCR machine. So you have heard already about the PCR because of this COVID thing. The PCR machine is able to magnify or multiply the cDNA into several copies. You can multiply the cDNA million times or more. The genomics technology is made possible by computer software, no? So nowadays, there is now a rapid automated DNA sequencing machine instrumental in the success of the Human Genome Project, which started in 1990 and ended in 2003. That enables the sequencing of the total human genome and was able to identify 30,000 genes in the human body. However, a genomic sequence is like a book. So imagine the human body has about 30 billion nucleotides, 30 billion letters of ATGC. A genomic sequence is like a book using an alphabet of only 4 letters without spaces or punctuation. Identifying the genes and their functions as a major challenge is now made possible because of bioinformatics. That is actually, the bioinformatics is the field in genetics where you use the computer as an instrument in order to identify the sequence. If you know the sequence, you can locate the sequence using computer. In bioinformatics, the genomic sequence is made possible to identify. The annotation of the genomic sequences at this level is one aspect of bioinformatics, which is defined broadly as the use of computers in the interpretation and management of the biological data available after DNA sequencing. So after DNA sequencing, you subject that sequence and let the computer identify the genome. If you know the sequence, you know the location of the sequence using the bioinformatics. So functional genomics. Genomic sequencing has made possible new approach to genetics called functional genomics that focuses on the genome-wide patterns of gene expression and the mechanism by which the gene expression is coordinated. So in genomic sequencing, it is now possible now to identify the functional genome and study which sequence of the DNA is functional and which is not using the computer. So there is what you call nowadays, there is the DNA microarray chip. For example, nowadays, to identify whether you have an asthma gene, they use microarray chip. It's a flat surface about the size of the postage stamp with up to 100,000 distinct spots. One spot accommodate one gene. So 100,000 distinct spots, each containing a different immobilized DNA sequence in order to use that for hybridization with the blood in order to identify whether the gene is present or absent. So supposing to diagnose you whether you have this particular type of disease, they use this microarray chip. Then that microarray chip has the specific gene for that specific protein. So, and then extract the DNA from your blood and then cut them into pieces and then put that in the microarray chip, and then using the hybridization technique, the DNA or RNA is then isolated on a growing condition, and then you can now identify whether or not the gene of interest is present, whether you have asthma or whether you have your genetic disease, G6PD, or you have your other enzyme deficiency can now be identified using the microarray chip. The DNA microarray are used to estimate the relative level of gene expression of each gene in the genome. So you can now identify. So this is an example of the microarray chip. One spot there could accommodate one gene of interest so that you can identify whether you have the defective gene, so or the gene of interest is present or absent. Whether the genetic defect is present or absent, you use this particular chip. No, one spot there is one gene. So this is the sequence. They now utilizes what you call reverse genetics. Mutation is reverse genetics. So what they do is identify the gene, they will destroy it, they will mutate it, and they will try to identify the effect of that particular mutation. So mutation has been traditionally provided the raw material needed for genetic analysis. The customary procedure has been to use a mutant phenotype to recognize a mutant gene and then identify the wild type or the normal function. So the recombinant DNA technology has made possible another approach often called reverse genetics, in which the wild type genes are cloned and then intentionally that gene is mutated or destroyed in specific ways. You can use radiation or introduce back into the organism and then find out the effects of that mutation. So in reverse genetics, they identify the normal gene and then they will subject that normal gene into mutation. And then that mutated gene is inserted back to the cell and try to find out the effect of that mutation. That is what you call reverse genetics. So in reverse genetics, you destroy the gene in order to find out the effect of the defective gene. So you call it reverse genetics. So identify the normal gene, you isolate that gene, you destroy it, you mutate it, and then insert the mutated gene, and then find out the effect. That is what you call reverse genetics. There is an example here. People that are unable, so after destroying the gene by mutation, people that are unable to remove the UV-induced lesion or damage are sensitive to sunlight. So that is one effect of the damaged gene using the mutated gene. So in bacteria E. coli that are unable to remove also the UV-induced DNA damage are also sensitive to sunlight. So this is what they found out. So they removed the gene for the sensitivity to light, and then they damage it using UV radiation. And then the people that are unable to remove the UV-induced lesions were found out to be sensitive to sunlight. So this is one method by which they identify the effect of radiation in the defective gene. And then therefore you can reconstruct another gene in order to correct the error. So nowadays, it is now possible to correct the error of our gene by inserting a new sequence into the damaged area of our genome. So the gene targeting, the procedure for introducing mutation into a specific gene is called gene targeting. The gene targeting in embryonic stem cells involves homologous recombination between target gene in vector and the target in the genome. So the target gene in the vector contains unrelated DNA so that recombination disrupts function of the target genes. So cells with targeted gene mutations can now be selected by including a selectable marker in the sequence that are incorporated into the genome in order to identify the gene of interest. So there is what you call a selectable marker. So they use a selectable marker. And so the gene that has been introduced to the animal is called transgenic animals. Those animals that have been modified by inserting the genome that are corrected or modified is called transgenic animal. So the germline transformation involves the insertion of the genes into reproductive cells like sperm cell or egg cell, which permanently alter the genetic content of the individual because the revised gene is inserted into the gonads, or it is inserted into the productive cells like sperm cell or egg cell. It is introduced there, then forever, that particular introduced gene will now be inherited forever. So transgenic organisms are used to study the functions of the specific genes in the development of the disease. So the transgenic animals, they use that for the study of the defective genes. So they use the animals in order to find out the effect of the altered gene using the radiation and find out the effect of that alteration, and later on study how to restore it to normal. So that is now the field in genomics. So the field in genomics enable us to insert the gene of interest in order to correct the error. So it is now possible that we can identify the defective gene using the DNA sequencer and then splice out that defective gene using the restriction enzyme and then insert a corrected DNA into the site. So that is now what you call genomics. So in genomics, it's now possible to insert a new DNA into the site and express it according to the particular function. So in applied genetic engineering, you can modify the plants already. Like for example, there is already a BT corn. There is a gene called BT. The BT gene is present in bacteria. And that particular gene resists the rotting of the plant. Therefore, they insert the BT gene into the corn or into the tomato. So the tomato will never rot because it has a gene already. So you can see here, yellow rice, because they have inserted the gene for the production of the vitamin carotenes. So they have engineered the carotene gene and they have inserted into the rice so that the rice becomes yellow. So animal growth rate can also be genetically engineered. So nowadays, you can insert a gene in order for the fish to rapidly grow. Engineered microbes can also help degrade the toxic waste product. So in mitigating pollution, they use already genomics in accelerating the degradation of the toxin using the proteins that are inserted into the microbes. The production of the useful proteins is a primary impetus for recombinant DNA, or is the primary impetus in science of genomics. So in biomedical applications, the application of genomics, recombinant DNA technology is used to produce large amount of important products, like for example, insulin. It used to be before insulin is produced by harvesting the pancreas of the animals. But nowadays, that is no longer practiced because bacteria can now multiply or can produce human insulin using the recombinant DNA technology, which is a subject in genomics. So animal viruses such as retroviruses are useful as vectors for the gene, because these RNA virus could now be used to insert the gene of interest. So supposing you have isolated already the gene, or supposing you have already reconstructed a correct DNA sequence, that correct DNA sequence can now be inserted into the retrovirus and let the virus insert it into our body. So this is called retroviral vectors in the gene therapy to treat single gene disorders. So recombinant DNA probes can detect genes that are defective, that are hereditary diseases. So nowadays, you can identify hereditary diseases using the DNA sequencing and bioinformatics. So what is done is get a DNA from your blood and identify, using the probe, identify whether or not you have defective gene for that particular disease. And then once you are detected that you have the defective gene, you can construct a new DNA sequence which will now be inserted back to you so that your inherited disease could be reverted. So major breakthrough in disease prevention would now come through the development of the synthetic vaccines produced by recombinant DNA technology. So nowadays, recombinant DNA technology is made possible because we can construct an artificial gene actually using the nucleotides that are available, the free nucleotides available. We can construct a new gene for our body because there is now a machine called gene constructor. There is a machine already available in the market that could enable to construct a DNA. So you can construct a DNA of interest using the nucleotides. So the nucleotides of our imorigine could be used for raw material for the construction of the genomics, the new genes of interest. Or you can construct a correct DNA using the nucleotides that are available. So we can reconstruct a recombinant DNA in our body in order to correct the error of the defective gene using what you call recombinant processes. Now, take note that recombinant DNA technology could be natural or could be artificial. Natural in a way that there are enzymes in our body that are able to correct the error by itself because, as I told you in my lecture before, our body is able to detect the errors of DNA sequences. And once the error are detected, our body has its own splicing enzyme that will remove the error and resynthesize the correct DNA sequence. That's why that is what we call natural recombinant DNA process. But there is an available artificial recombinant DNA process wherein if you have identified the defective gene, we can construct an artificial DNA with the correct sequence and insert it into the area where the damage is in order to reverse the genetic disease. So that ends my lecture today, and that is the end of my lecture. Thank you very much and good morning.